Hi students, greeting of the day and welcome to my video channel. Here we are going to deal into this portion of video with the important questions of board examinations. Since the exams are coming and it's a pressure on you all how to solve the questions, and there were plenty of emails, and as per those emails what we have planned is our group has taken out the questions as per the marking scheme of chemistry paper for 2019, and we are dealing with the answers of those questions so that it becomes easier for you to write the answers. Just an example to it, if suppose the solid state, the first chapter, has one question of 1 mark and one question of 2 mark. So here how much is to be written in 1 mark? So for that we have done the homework that 70 marks paper is there and there are around 30 questions. 1 mark counts for around 2 minutes because you require the time to check the paper as well as you require the time to make the corrections. So 1 mark question is to be written in 2 minutes, and those questions as per the marking schemes have been dealt into different chapters, and that is just a exercise which you have to do. Now take up the questions of 1 mark of the first chapter, read the questions, read the answers, memorize the answers, and then give time to write the answers on practical basis. And be cautious students, take care of the time. If the time limit is to be considered properly, then at the time of examination all the questions will be solved and there won't be any question which will be left by you. So practice these questions properly, and this is an effort which is made for you, taking into account the 29 years of my experience with CBSE students and IIT experience. So take care that you solve the questions properly and be positive in answering the questions. Same way you write the answers as you are going to write in the examination. It's like a examination before the examination. So take care of it and prepare with it. Thanks a lot.
This is 14th chapter of Biomolecules, and as per the marking scheme provided by CBSE for 2019, there will be 2 questions of 2 mark, that is 4 mark questions will be asked. So we are dealing with 2 mark questions. The first question says the 2 strands of DNA are not identical but are complementary. Explain. The DNA consists of 2 strands of polynucleotides coiled around each other in the form of a double helix. The nucleotides making up each strand of DNA are connected by phosphodiester bonds. These forms the backbone of each DNA. DNA is deoxyribonucleic acid strand from which the bases extend. The bases of one strand of DNA are paired with bases of another strand by means of hydrogen bonding. This hydrogen bonding is very specific as the structures of bases permit only one mode of pairing. Adenine pairs only thymine through 2 hydrogen bonds, and guanine pairs with cytosine through 3 hydrogen bonds. The 2 strands of DNA are said to be complementary to each other in sense that the sequence of the bases in one strand automatically determines that of the other. These strands are not identical but complementary. Question number 2. How can reducing and non-reducing sugars be distinguished? Mention the structural feature characteristic reducing sugars. Those carbohydrates which reduce Tollen's reagent to give silver mirror to form brick red precipitate with Fehling solution are called reducing sugars, whereas those which do not are non-reducing sugars. All monosacharides are reducing sugars due to the presence of aldehyde group or alpha ketone, which you can see. Those disaccharides like sucrose are not reducing sugar because aldehydic and ketonic groups are linked and not free. All polysaccharides are not reducing. Question number 3. Amino acids may be acidic, alkaline or neutral. How does this happen? What are essential and non-essential amino acids? Name one of each type. Acidic amino acids, those amino acids which contain two carboxylic group and one amino group are called acidic amino acids. Example, aspartic acid. Basic or alkaline amino acids, those amino acids which contain two amino groups and one carboxyl group are called basic amino acids. Example, arginine. Neutral amino acids, those amino acids which contain one amino group and one carboxyl group are called neutral amino acids. Example, glycine. Essential amino acids, those amino acids which are not synthesized by our body are called essential amino acids. They must be part of our diet. Their deficiency leads to the disease such as kwashiorkor, water balance in the body is disturbed, and example is leucine. Non-essential amino acids, they are synthesized by our body. They are also called dispersed amino acids. Example, serine. Question number 4. What is structural difference between the nucleoside and a nucleotide? Second part, the two strands of DNA are not identical but are complementary. Explain. Nucleoside contains ribose and deoxyribose sugars and heterocyclic base. Example, adenosine. Whereas nucleotide contains phosphoric acid residue also along with heterocyclic base and pentose sugar, that is adenosine triphosphate ATP. Second part, DNA consists of two strands of polynucleic coiled around each other in the form of double helix. The nucleotides making up each strand of DNA are connected by phosphodiester bond. These forms the backbone of each DNA strand from which the base extend. The bases of one strand of DNA are paired with bases of another strand by means of hydrogen bonding. This hydrogen bonding is very specific as the structure of bases permit only one mode of pairing. Adenine pairs with thymine through the hydrogen bond and guanine pairs with cytosine through 3 hydrogen bonds. The two strands of DNA are said to be complementary to each other in sense that the sequence of bases in one strand automatically determines that of the other. These strands are not identical but complementary. Question number 5. A part, write the important structure difference between DNA and RNA, and B, mention the names of the bases produced by hydrolysis of DNA. So the first part A, DNA and RNA, we have to distinguish between them. The first one of DNA, it has double helix structure, while RNA, it has single helix structure. DNA, it has adenine, thymine, guanine and cytosine, while RNA, it has adenine, guanine, cytosine and uracil. B part says the hydrolysis of DNA gives adenine, thymine, guanine and cytosine. Question number 6. Name the chemical components which constitute nucleotides. Write any 4 functions of nucleotides in a cell. Nucleotides are made up of heterocyclic base containing nitrogen, 5 carbon sugar and a phosphate unit or group. AMP, adenosine monophosphate, ADP adenosine diphosphate and ATP adenosine triphosphate. Functions. They act as energy carriers. Second, they are monomers of nucleic acid, that is DNA and RNA. Third, they store and pass on hereditary characteristic. Fourth, they synthesize protein. Seventh question, what happens when D-glucose is treated with following reagent? First HI, second bromine water, third HNO3. The first one, answer to first one is the glucose, CHO, CHOH whole 4, CH2OH. When made to react with HI in excess, it forms CH3, CH2 whole 4, CH3, N-hexane and 6 water molecules are eliminated. The second one, glucose, CHO, CHOH whole 4, CH2OH. When made to react with bromine, it gives COOH, CHOH whole 4, CH2OH, gluconic acid plus HBr. And third one is glucose, that is CHO, CHOH whole 4, CH2OH plus HNO3 gives COH, CHOH whole 4, COH, saccharic acid. Question number 8. The particles in nucleus of the cell responsible for heredity are called chromosomes, which are made up of proteins and another type of biomolecules called nucleic acids. DNA, deoxyribonucleic acid and RNA, ribonucleic acid are long chain polymers of nucleotides which are made up of nitrogen containing heterocyclic bases, a pentose sugar and a phosphoric acid residue. What are the two types of RNA? This is first part. Second part, what is DNA fingerprinting? And third, give uses of DNA fingerprinting. So answer to first part is messenger RNA, m-RNA and transfer RNA, tRNA. They are the two types. Then second, the sequence of the bases of DNA is unique for each person, and information regarding a person with the help of DNA is called DNA fingerprinting. Third, it's used in forensic labs for identification of criminals. B, it is done to determine paternity of an individual. Recently, it has been used in case of Rohit Shekhar, who is the son of Narayan Dutt Tiwari, a politician. C, is used to identify the dead bodies by matching their DNA with parents. D, is used to identify racial groups to rewrite their biological evolution. Question number 9. First part, give one structural difference between amylose and amylopectin. Second, name the protein and their shape present in oxygen carrier in human body. Third, name two fat storing tissues in human body. Answer to first one is amylose is straight chain polymer of alpha D positive glucose, whereas amylopectin is branch chain polymer of alpha D glucose. Amylose is water soluble component 15 to 20%, whereas amylopectin is water soluble component 80 to 85% of starch. Hemoglobin is globular protein and is spherical in shape. Third, A, liver. B, adipose tissues. Question number 10. Explain the following terms: mutarotation and avitarinosis. Mutarotation, answer to it is the spontaneous change in specific rotation of an optical active compound is called mutarotation when it is dissolved in water. When glucose is crystallized from the concentrated solution of 30 degrees Celsius, it gives glucose having alpha, that is plus 111 degree. Specific rotation changes to 52.5 degrees when it is dissolved in water. It is because alpha glucose changes into beta glucose. This change in optical rotation is called mutarotation. Multiple deficiencies caused by lack of more than one vitamin in human being is called avitaminosis. Avitaminosis. Question number 11. Answer the following questions briefly. What are reducing sugars? First part. Second part, what is meant by denaturation of protein? Third, how is oxygen replaced in our atmosphere? First part, those carbohydrates which have a free aldehyde group and reduced Fehling solution are called reducing sugars. Second, when tertiary and secondary structures of protein is ruptured by heating, change in pH or any adding electrolysis is called denaturation of protein, where the nature of the protein changes, and when the nature changes, it becomes difficult for the protein to have that impact. Third, we take oxygen from atmosphere and reduce CO2. Plants like CO2 and water forms from atmosphere to prepare their food in presence of sunlight and release oxygen, and thus oxygen is replenished in our atmosphere. Question number 12. Answer the following questions briefly. First part, what are any two good sources of vitamin A? And second part, what are nucleotides? So the first part is carrot and cod liver oil. They are the good sources of vitamin A. And B, nucleosides are monomers of nucleic acid. They consist of heterocyclic base, pentose sugar and phosphoric acid residue. Second, A, monosaccharides. B, oligosaccharide. C, polysaccharides. Question number 13. Define the following terms: coenzymes, mutarotation in biomolecules and nucleosides. So the first one, coenzymes, those prosthetic groups which can attach to enzymes at the time of reactions are called coenzymes. They increase the activity of enzymes. Mutarotation in biomolecules is the second one. A difference in the simple base of DNA molecules can cause change in amino acid sequence, which leads to mutarotation. Nucleotides, they are monomers of nucleic acid. They consist of heterocyclic bases, pentose sugar and phosphoric acid residue. So these are the questions which we had to deal in this chapter, biomolecules. So that is of two questions of 2 marks, and 4 mark questions will be asked. So best of luck for your examination. Have a proper practice, have a proper diet, have a proper sleep. So take care of yourself and bye.